Hello students, welcome back to your own channel Vedantu NEET English. This is BASAVARAJ sir, your biology master teacher. So students, today's class is going to be one of the high weightage chapters, that is principles of inheritance of inheritance chapter. Now in this chapter, let me tell you, it has been broadly classified into two categories, and the first category is just the theory part, and the second category is going to be the entire numericals. So that's how which I'm going to deal. And we will be dividing the entire big chapter into two parts, part 1, part 2, and after part 1 and part 2 we'll do a part 3 which will only have numericals. Okay, so we'll go in such a easier fashion, because let me tell you why. Once we do such bifurcation, right, such division in the numericals part, all of you can ask me doubts and your doubts can be cleared live. So let's have a proper dedicated class for numericals, and that is separate. And first we will learn the theory behind every single concept, and today is the part 1 of that theory. Okay, part 1 of the theory. So students, let me know in the chat if I am audible and the beautiful DNA behind me is visible to all of you. Quickly let me know in the chat if I am audible and visible to all of you. Yes, audible and visible. Yes, I can see green hearts in the chat, which makes me happy. Obviously, all the green hearts in the chat. Yes. Hello Meghna, hello Flamingo, hello Ravindrani, hello Madhuri, Meghna, Satvi, Tanuja. Yes, hello. So welcome back to the channel. Now students, I want quickly all of you to mark your attendance, and we will get to the class now. Now students, all of you are watching it recorded. I would also want you to smash that like button, but mark your attendance, which obviously help us as a teacher here for us to grow, right? For us to grow. Now students, let's go with the basics, right? All the way from the basics, and we will not rush anything. I will make sure you understand every single point of this chapter. It all the way from the basics. When I say basics, I mean what is the meaning of dominant, what is the meaning of recessive, what is a modified type, what is a wild type in that, right? What is a phenotype, what is the genotype, what is homozygous, what is heterozygous, what is the meaning of a character, what is the meaning of a trait, what is the meaning of a gene, and what is the meaning of allele. So all the way from the basics. Even if you do not know a single point of this chapter, you do not even know anything in this chapter, after learning my class, you will be able to solve all the questions from this chapter. That is my guarantee to all of you. Okay, so students, can we start the class today with all the way from the basics in a much more chill and informative manner. Now students, I want you to understand, first of all, what is the meaning of genetics? What is the meaning of genetics? Students, genetics is a branch of biology. It is a branch of biology that deals with inheritance or heredity and variation. It deals with heredity and variation. It is a branch of biology. Yes, it is a branch of biology that deals with heredity and variations. Okay, it deals with heredity and variations. That is the meaning of genetics. Okay, now students, how many of you have heard in your life that your eyes look like your mother's eyes? How many of you have heard in your life your nose is just like your father, you're tall as your father, you're beautiful as your mother? Or have you ever heard of this, when they compare you to your parents, the way you look, the way you talk, sometimes the way you represent yourself? Have they compared in your life? You might be, some or other way, the relatives have told you this, right? They have not told you this. Now students, what is the meaning of all this? The meaning of all this is, there's a beautiful word called as inheritance. What is the meaning of inheritance, students? Inheritance. The beautiful word called as inheritance. Inheritance is nothing but it is a process. It is a process by which genetic characters are passed from parents to the offspring. from parents to the offspring, genetic characters or traits are passed from parents to offspring. Now, how many of you have you ever seen in the wild a cat giving birth to a dog giving birth to a cat? Does it ever happen? No. So there is a beautiful word called as like begets like. That is transfer of characters from parent to the offspring. Obviously, a dog will give birth to a puppy, a cat will give birth to a kitten, a human will give birth to a human baby. So that is called as inheritance. That is called as inheritance. Any doubt there? Quickly ask me in the chat. That is called as inheritance. Now, all of you told me, sir, yes, I am almost similar to my parents. Yes or no? Right, you are almost similar to your parents. But are you the exact copy of your parents? Tell me. Are you an exact copy of your parents? You will be like, no, sir. There are some changes, right? My nose is like my father, but my eyes are like my mother. My cheeks are a little different. My arms are a little different. So, are there some changes from parent to the offspring? Yes. Now, those changes, right? Those changes are also called as variations. Those changes are also called as variations, students. That is very important. The changes is called as variations. It's called as variations, students. Variations. So in this chapter, in today's chapter, we'll be learning about inheritance, that is transfer of genetic information from the parent to the offspring, and also some variations which are seen when the information is being passed. There are small changes called as variations, and that is your entire chapter. That is your entire chapter. So with that being said, students, can we start today's amazing class? Can we start the class, students? Yes. It's a very easy class because I will be teaching you from all the way from the basics. When I say basics, I mean character and a trait. What is a character and what is a trait? Now, if I give you the example, it becomes very clear. But before we go to the example, let's understand the definition of a character now, shall we? What is a character? It is the feature of an individual. Basically, character is nothing but it is a feature of an individual. It's a feature of a individual. Now, when I say feature, I mean phenotypic feature. Mostly phenotypic feature. Yes, I mean phenotypic feature here. Okay, phenotypic property or phenotypic feature, that is a character. A quick example will help you. A quick example is going to be height, color of a flower. Color of a flower is also a character. Yes, height of a plant is a character. Shape of a seed is also called as character. It is basically a feature of an individual. Height is a feature, color of a flower is a feature, shape of the seed is a feature. But is there any variations mentioned here? It is something called as variations? No. Now, variations will come in when we talk about the trait. When we talk about the trait, friends, the trait is nothing but the characters plus the variance. Understand the example. The example here is very simple, students. Here I spoke about height. Here I'll talk about its variation. That is the plant tall or is it a dwarf plant? Here I talk about color of the flower, but here I will talk about the variance. Is it white flower or is it a red flower or violet flower? Here I spoke about shape of the seed. Is the shape of the seed round or is it wrinkled? Students, do you understand now? Students? This is called as a character, that is a feature of an individual, mostly phenotypic. Here it is a character plus variance, that becomes a trait here. For example, here we have height, color of the flower, shape of the seed. But when we come to the trait, we talk about the specific trait variants, that is tall or dwarf, white or red, color of the shape, that is shape of the seed, round or wrinkled. Any doubt here? Quickly ask me, students. Any doubt here? No, right? Very simple. This is the basic things which every student attempting to understand genetics should know. Okay, should know. For example, students, for example, what is the difference between a gene and an allele? What is the gene, students? What is the gene? Now, all of you will tell me, sir, gene is the unit of heredity. That is true. But if you want to talk about the actual definition wise, gene is nothing but a small segment of a DNA. Let me teach you. Let me teach you. Listen to me. Listen to me. Just listen to me, students. Gene. For example, students, this is a DNA. This is a piece of DNA. From this piece of DNA, students, from this piece of DNA, a small segment, if you take a small segment, the small segment, that becomes a gene. That becomes a gene. Gene is the smallest segment of DNA actually. It is the smallest segment. It is going to be the smallest segment of DNA. Smallest segment of DNA. Deoxyribonucleic acid. Smallest segment of a DNA. Okay, that is a gene. That is a gene now. So all of you know what is a gene now. All of you know what is a gene. Then what is an allele then? What is an allele? Students, how many of you know what is twins? How many of you know what is twins? Twins are basically the two humans. Sometimes they are similar, they look alike. Sometimes they don't look alike. That is also true. But twins are the two humans, right, which are born from the same mother obviously and same zygote and everything. That I'll tell you in another class. But twins are the organism or humans which look alike, right? Now, can I call this? There is a gene here. This is a gene. Allele is like a twin. Allele is like a twin. Allele is nothing but alternative form of a gene. It is going to be alternative form of a gene. So a gene will have two different twins. A gene will have two different twins. That is an allele. Now, if I can ask you one higher level question now. Is it always the case one gene will always have two alleles? True or false? Tell me in the chat. Do you think a particular gene will always have two alleles? True or false in the chat? Is it true? A gene will have always two alleles. True or false? A gene will always have two alleles. Tell me. False. Obviously false. Because there is a concept called as multiple allelism, which we'll be talking in today's class. There's a concept called as multiple allelism. Multiple allelism, in which a single gene will have multiple alleles. A single gene will have multiple alleles. That is called as multiple allelism, and that concept is basically the backbone of blood grouping. Backbone of ABO blood grouping. ABO blood grouping. So yes, a gene can have multiple different alleles. It is possible. It is possible. Okay. Now that is an allele. The next concept is what is a phenotype and what is a genotype. What is a phenotype and what is a genotype? Students, it's very simple. Phenotype is basically the external morphology and genotype is the genetic constitution. Right? Phenotype is nothing but observable morphology. Phenotype is nothing but observable morphological appearance. Observable morphological appearance is called as phenotype. For example, a tall plant, a dwarf plant, that is the observable feature. That is, you can look at the plant and tell, yes, this is a short plant or this is a tall plant. Color of the flower, yes, flower color. If you look at the flower itself, you will be able to tell, yes, it is a red color. It is a red color flower. Now what is the genotype, students? Genotype is the genetic makeup. It is going to be the genetic makeup. For example, capital T, small t, capital T, small t. This is the genetic makeup, also called as the genotype. Also called as the genotype. Is that point clear to all of you? Yes or no in the chat quickly. Is that point clear to all of you? This is called as phenotype. This is called as genotype. Now, what is the one more concept? The other concept is called as homozygous and heterozygous. Homozygous as well as heterozygous. Very simple again. Very simple. Very simple. Very simple. Now, I spoke about twins. For example, Ramesh and Suresh. Now, let's take someone from the chat now. Divya and Meghana. Okay. Divya and Meghana are the two twins here. Now, if Divya and Meghana look alike, that is, they look very same to each other, you cannot tell who is Divya, who is Meghana. You cannot tell. That is identical twins, and that is also an example of homozygous condition. In the case of homozygous conditions, the alleles look alike. The alleles look similar. The two alleles look similar. The two allele looks similar. For example, students, for example, capital T or small t. This is called as homozygous dominant. This is called as homozygous recessive. Homozygous recessive. Okay. Now, what is a heterozygous condition? For example, if Divya and Meghana do not look alike, they look very different. Divya has blue eyes and Meghana has red eyes. Can you imagine? So they do not look alike. They do not look alike. That is, the two alleles look different. The two alleles look different here. They look different. For example, students, capital T, small t. Capital T, small t. This condition is called as heterozygous dominant. It is called as heterozygous dominant. Done? Done? Yes. This is homozygous and this is heterozygous condition. Now, I spoke about dominant and I spoke about recessive. I spoke about dominant and I spoke about recessive. Now, what is this dominant and what is this recessive now? Very simple again, very simple. Dominant is basically allele which influences the phenotype. The allele which influences the phenotype. Which influences the phenotype. Even this allele which influences the phenotype, even in the presence of alternative allele. Even in the presence of alternative allele. Even in the presence of alternative allele, it can express itself. Okay, it can express itself. That is called as dominant allele. This is like the loud kid in the class, whatever happens, this kid will keep on talking. Blah, 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 shouting, keep shouting from the back. Okay, loud kid in the class who will always keep on talking. That is called as dominant. And here in this case, a allele which is able to express itself even in the presence of alternative allele, that is called as dominant allele. For example, students, here we have capital T, small t. Here we have capital T, dominant alleles. Capital T, small t, this is the dominant allele because this allele is able to express even in the presence of alternative allele. This small t is the alternative allele. Even in the presence of alternative allele, capital T is able to express. The capital T is able to express. Okay, capital T is able to express. Now students, now. What is the meaning of recessive allele? What is the meaning of recessive allele? Recessive allele is the quiet kid in the class. The quiet kid will only talk when no one is around, right? The quiet kid will only talk in the class when no one is around. So that is a recessive allele. Recessive allele is an allele that influences the phenotype. That influences the allele which influences the phenotype, which influences the phenotype only in the absence of absence, only in the absence of dominant allele. So whenever the dominant allele is absent, only when the dominant allele is absent, recessive allele is able to express. Recessive allele is able to express. Okay. Now for example, this particular allele will only express in this condition, small t condition. It is able to express only in the homozygous recessive condition. Homozygous recessive condition. Yes. Is the point clear to all of you now, students? Is the point clear to all of you? Now students, in the examination, they can ask you some, instead of telling you, sell the unit of life Monday, sell the unit of life on Monday. Okay. So because we are balancing, right, we are balancing RNA as well as line by line. Both are equally important. See this here you learn the in detail concept. In line by line you revise and learn again. Right, you realize we have to go in hand in hand, right? You have to go in hand in hand. Now students, in examination, sometimes they can call dominant allele as wild type, and recessive allele as, sometimes they can be mentioned recessive allele as modified type. Okay, remember these two words also, the alternative names to the dominant as well as recessive alleles. Clear? Yes? Clear all of you? Now students, now let's go to the next concept. That is going to be Mendel now. Finally we come to Mendel. Our uncle Mendel is here now, and we'll write on the few beautiful points about Mendel and what was his work, when was he born and all that, and let's see what we get in the end of it. Okay, so I want everyone to open your books right now. Tamil, no, I can understand Tamil, not speak Tamil, I can speak Kannada. Kannada is my mother tongue, right? So now we will learn about Mendel one by one. Okay, now students, first of all, Gregor Mendel. Let's talk about Gregor Mendel. Gregor Mendel was born on your July, last month. Okay, last month of 1822. Okay, July 22nd, he was born on. Okay, he was born on July 22nd, 1822. He was born on July 22nd, 1822. Okay. Now, what do you think Gregor Mendel worked on? What was his experimental animal or the plant? Tell me. Which particular organism did Gregor Mendel work on? Yes. On which particular plant or animal he worked on? His main work was on a plant which you use in your matar paneer. You know, you make paneer gravy, right? In paneer, you add something called as green small matar, which is in Hindi. That is called as your pea plant. And scientific name is Pisum sativum. Pisum sativum. Pisum sativum. Okay. Pisum sativum. Okay. Pisum sativum. Pisum sativum. Now students, you need to understand what was his main work, as in like, see, he worked on pea plant, but what is his work called as? What is his work known as? His work is known as something called as hybridization. His work on the pea plant is called as hybridization. It's called as hybridization. Now, what is a hybridization? There are multiple steps in hybridization which I will teach you later. First, for example, there is first step is collecting of parents, crossing over, then selfing of F1. All that we will learn in today's class only. Okay, all that we will learn in today's class only. Okay. Now, the next concept which you need to understand here is how long did you work on the exam and on this experiment? For example, you are studying class 10th for 1 year, class 12th for 1 year. If you take a drop year, 1 year or 2 year drop year, right? How many years do you think Mendel worked on his pea plants? Just take a wild guess. How many years did he work on? He worked from 1856, students, 1856 to 1863. 1856 to 1863. 3. Now guess how many years is that now? How many years is that? He worked for 7 long years. 7 long years, students. 7 long years. 7 long years, that is. He worked on pea plant for 7 years. That's a lot of time. That is a lots and lots of time, friends. 7 years. Okay, 7 long years on the pea plant. Okay. Now the next question arises. He took a pea plant. Okay, he took a pea plant. Now, what do you think he found in the pea plant? Now, in the pea plant, he had to observe something, right? He had to observe something on the pea plant. So, he observed 7 different characters and 14 different traits. He observed 7 different characters. He observed 7 characters. 7 characters and 14 traits. 14 traits. 7 characters and 14 traits. Now students, now why was Mendel different? The Mendel was a little different apart from his other competitors is because his main work in biology also used mathematics. He also used mathematics and statistics in the biology. That was one of the key distinguishing features of Mendel. Now before we go to that, you need to understand one more thing. Which type of plant did he take? Which type of plant did he take? That is very important. That's very important. He selected 14 true breeding lines. True breeding lines. Now what is this true breeding line, students? True breeding lines is nothing but plants which has undergone repeated self-division. So repeated self-pollination. Repeated self-pollination. Repeated self-pollination. Repeated self-pollination. They keep on self-pollinating, and we end up getting true breeding lines. And remember students, these true breeding lines are homozygous in nature. Homozygous in nature. They're homozygous in nature. Is that point clear to all of you? Okay, so he used maths and statistics also. He used mathematics and statistics. He used maths and stats. Okay, he used maths and statistics. Now one more thing which you need to understand here is the sample size. One of the key reasons why Mendel's work was such success is because he used very large sample size. Very large sample size. The meaning of sample size is that his experiment was on multiple plants. See, if you do the experiment on 1 or 2 plants and you end up getting the result, and there is a very high chance that the result might be false positive. Very high chance. So if you want your work, the research work to be more validity, you need to take a very large number of plants to experiment on, and that is called as a sample size. Large sample size. Large sample size. Now Mendel is clear now? That is about your Mendel. That is about your Mendel. Okay. Now here are all the different types of characters and traits which Mendel took into consideration. Okay. For example, students, how will the question be asked here? It's very simple. Question they'll ask you, simple question they'll ask you is, how many characters did he take? What is the answer now? How many characters did Mendel take? You will tell, all of you will tell me, sir, 7 characters he took. Okay. Here. 7 different characters. Now, how many traits did he take? All of you will tell me, sir, 14 traits. Now, they can twist the question they can ask you. Now. They will give you a list of different traits. And then they will ask you, how many of the following are dominant? Beautiful question now. They will give you multiple traits and they'll ask you how many of the following are dominant, or they can directly ask you which of the following is dominant. Simple. So you'll be stuck that time. Now do you want a trick? This is not my trick. This is a trick which I have taken from the internet, and the trick is very simple. The trick is very simple. The trick is called as gravity. The trick is called as gravity. The trick is called as gravity. Now students, here you need to focus now. Now what is this G? The G stands for green. G stands for green. That is going to be your pod color. That is going to be your pod color. Can you see? Pod color. The G is going to be green. That is going to be pod color. So write down that also. That is going to be your pod color. Pod color. Okay. Now what is going to be R? R is nothing but your round. R is nothing but round. R is nothing but round. Round here is going to be what? Round here is going to be your seed shape. Round here is going to be seed shape. Seed shape. Round is going to be seed shape. Then we have A. Then we have A, students. Then we have A. What is this A? A is nothing but axial. A is nothing but axial. Axial. Axial is nothing but axial. Here is nothing but here. Can you see? Axial. Okay, 1 second. Axial. Axial, here it's going to be your position of flower. Flower position. Can you see here? That is going to be your flower position. Flower position. Flower position. Now next V. What is V now, students? What is going to be the V? Here. V is going to be nothing but violet. Where is it? Violet here. Here. Flower color. Here. That is going to be your flower color. Can you see? Flower color. Okay. That is going to be flower color. Violet, right? Violet, yeah. And that is going to be your flower color. Flower color. Now what is I then? What is I? I is nothing but inflated. I here is inflated. I inflated or full. Inflated or full. Here, can you see? Inflated or full. That is pod shape. That is going to be pod shape. Pod shape. Done. Now what is T? T is nothing but tall. T is nothing but tall. What is tall? That is plant height. That is going to be plant height. Plant height. Plant height. Now what is Y? What is going to be the Y here, students? What is going to be the Y here? See here. Y is going to be your, where is it? Yellow. Seed color. That is going to be yellow. That is going to be yellow. Okay. That is going to be your seed color. That is going to be seed color. Seed color. Now with that, students, with that they can ask you multiple questions. They can ask you multiple questions. Now they can give you any of the following dominant trait, any of the following dominant trait. They'll give you, for example, in an option they'll give you 2 of them or 3 of them or 5 of them, and they'll ask you how many of them are dominant here, and the options will be 1 or 5 or 6 or 4. You understand? That's how the question can come, and you need to be prepared for such questions. Any doubt here, quickly ask me. Any doubt here, quickly ask me. No doubt, right? No doubt? Yes. Now with that, let's go to the next concept now. Let's go to the next concept. That is going to be luck. Now students, do you think luck is required? Do you think luck is required in experiment? What is pod? Pod is nothing but here. Pod is nothing but here, in which all the seeds will be present. In which, or the fruit, type of fruit only, in which all the seeds will be present. Have you not seen the pod? You might have seen the pod, right? You might have seen the pod, right? Now students, the answer is yes. You might think that luck is not required, but if Mendel was not lucky, we would not be studying any of his experiments now. So luck is important, students. Luck is important to some extent. Yes, luck is important. Yes, luck is important. Now let me teach you why. Let me tell you why. Now, do you think Mendel only worked on pea plant? Do you think the Mendel only worked on pea plants? Obviously not. Mendel also worked on other plants also. Mendel, our uncle, he worked on other plants also. For example, students, he worked on something called as hawkweed and beans. He also worked on hawkweed and beans. But when he worked on hawkweed and beans, the result was not same. The result which he obtained from pea plant, the results which he obtained from pea plants and the results which he obtained in hawkweed and beans, they were not same. He did not observe the dominance, he did not observe the law of independent assortment and all that here. So luck is very important. For example, students, what if Gregor Mendel ended up taking hawkweed and beans in the first attempt? Obviously then he would not have come to the same conclusion which we have right now. So luck is important, but let me tell you why he became lucky also. Because in the case of pea plant, out of the 7 characters, all the 7 characters which he took in the pea plant were present on 4 different chromosomes. They were present on 4 different chromosomes. He became lucky. That is 1, 4, 5, 7. Chromosome number 1, 4, 5 and 7. Now imagine if he ended up taking, hawkweed, but the chromosomes are not very far. No same result. Now since he took pea plant and all the different characters are there in 4 different chromosomes, he could observe the crossing over, he could observe the variations and everything. Okay, so luck is important to some extent, but let me tell you, luck is what you create. This is very true. So imagine if he did not do any work and just because of lucky was he able to do everything? No, he worked for 7 years. So luck will come for those who work hard. That is very important to understand. Yes, Mendel was lucky. But remember, students, luck will only come to students who work hard. You cannot expect to, become lucky and clear NEET examination without studying. That will never happen. Because Mendel worked for 7 long years in order to be lucky in his life. And unfortunately, students, his work was never even, given importance until his death. So luck is very important. Yes, he was lucky and unlucky at the same time. So if you work hard, you will also be lucky. Okay? Now, let's go to the next concept now. Next concept is very simple. Why did he select pea plant? It is very important to understand. Why did he select the pea plant? Okay. Obviously, easy to grow and maintain. Students, question will come. Here also, do you think it's very simple? Question will come from here. Right? It is very easy to grow and maintain. Yes. It's an annual plant. What is the meaning of annual plant? Its life cycle gets over in only 1 year. Right? Life cycle gets over in 1 year. What is biennial? 2 years. Perennial? Many years. Okay. Now, exhibits a variety of visible characters with contrasting traits. Yes. 7 characters, 14 traits. Right? Produces bisexual flowers. Yes. It is easy for cross-pollination then. Easy for pollination basically. Right? Pollination. Self-fertilizing plant and hence mating can be controlled easily. See, mating can be controlled easily. Self-pollinating plants, remove the anther, control the pollination. Also possible to conduct cross-pollination, I told you already. Produces a large number of seeds which you can use for plantation. It is a very cheap plant also. Not very difficult to find it. Annual means only 1 year. So students, do you realize why he took the pea plant? All of the following reasons. Any doubt here, quickly ask me, students. And students, quickly smash that like button. Okay, it's very important because you know that's how YouTube algorithm works. For teachers like me and everything, and even though how much ever we work hard, unless students show your support from your side, this video will never reach to all the students unless you show support. Right? That's why we always ask you. Why do you think we always ask you to like the video and comment like how the session is? The only when you comment on the video, other students will, without even seeing my lecture, they will realize, yes, this teacher might be good and this teacher will help me. Right? That's why we always ask you. Right? Now, next thing is the other names which it will be rarely asked, but you should know this. That is going to be a father of genetics, Gregor John Mendel. Then we have modern genetics, that is Batson. Then we have experimental genetics. Experimental genetics is nothing but T.H. Morgan. Experimental genetics is nothing but T.H. Morgan. And T.H. Morgan uncle will come later on. T.H. Morgan will come later on, and we will learn about something called as Drosophila melanogaster there. Drosophila melanogaster there. Okay, so don't worry, everything is going to come. Okay, it's a very easy chapter. It's a very easy chapter, and I will make sure you understand every bit of it. Okay, now, next thing is hybridization. Next thing is going to be hybridization. And as I mentioned earlier, there are three main steps in hybridization. The first step is collection of parents, right? We need parents for hybridization. First step is collection of parents. Second step is cross-pollination. First one is collection of parents. Collection of parents. Yes, collection of parents, or can I say selection of parents? That is better to understand. Selection of parents, that is better to write. Selection of parents. Selection of parents. And remember students, they should be true breeding lines. They should be true breeding lines. True breeding lines, always. True breeding lines, always. Now second step is cross-pollination. Cross-pollination. Second step is going to be cross-pollination. Third step is going to be selfing of F1. Selfing of F1 plants. Selfing of F1 plants is going to be your third stage. Three steps in the case of your entire hybridization. One by one we'll go, and it is very easy to understand now. Now, before we go to the crosses ahead, can I ask all of you a simple question now? Can I ask you all of you a simple question? The question is that in which condition or in which case do you find phenotypic ratio is equal to genotypic ratio? Option number A is going to be codominance. Option number 2 is going to be superdominance. Superdominance. Option number 3 is going to be incomplete dominance. Incomplete dominance. And option number 4 is going to be all of the above. All of the above. Tell me, in which of the following do you find phenotypic ratio is equal to genotypic ratio? Go. Let's see the answer. Let's see the answer. Phenotypic ratio is going to be equal to genotypic ratio. That happens in the case of your incomplete dominance. Nice. It happens in the case of incomplete dominance. Okay. Now students, can we do the monohybrid cross now? So with that being said, can we do the monohybrid cross? We will do it in a much more simpler manner, so everything will be able to understand, and there should not be any doubt after this. There should not be any doubt after this. Okay. So inheritance of one gene, also called as your monohybrid cross. It is also called as what? Monohybrid cross. Monohybrid cross. Okay. Monohybrid cross. So starting off, we need first step is what, students? First step in hybridization is selection of parents. Yes. First step is called as selection of parents. So we will be taking homozygous dominant, homozygous. One second, students. Let me write this side. We will be taking homozygous dominant plant, homozygous dominant here. Yes, homozygous dominant, and we'll take homozygous recessive. We'll also take homozygous recessive plant. Now tell me, homozygous dominant will be capital T, capital T. Homozygous recessive will be small t, small t. Okay, small t, small t. This is basically the parents. This is selection of parents. Now, what is the second step, students? The second step is going to be cross pollination. Cross pollination, yes. But before that, we need gametes. We need gametes. Gametes are here. This is the gametes. Gametes. Now we have crossing over. Crossing over. We end up getting capital T, small t. This right here is the F1 plant. This right here is the F1 plant here. F1, capital T, small t. Now tell me, this capital T, small t, what is going to be its phenotype? The phenotype is going to be a tall plant. Tall plant. What is the genotype here? The genotype is heterozygous dominant. Heterozygous dominant. That is going to be the genotype. Phenotype and genotype. Done. That is no. Now, what is the third step, students? The third step is something called as selfing of F1. Selfing of F1. Selfing of F1. Now, what do we do in selfing of F1? We take capital T, small t, crossing with capital T, small t. Yes, that's how we cross it, and we end up getting the gametes. So let me do the cross here. Let me do the cross this side. Let me do the cross this side. Okay, this is called as Punnett square. This is called as Punnett square. Okay, Punnett square. Punnett square. Now, what are the gametes? Capital T, small t, capital T, small t. Now, what do we get here? Homozygous dominant, heterozygous dominant, heterozygous dominant, and homozygous recessive. Homozygous recessive. Now, students, I want you to tell me the phenotypic ratio as well as the genotypic ratio now. Phenotypic ratio as well as the genotypic ratio. First we'll write phenotypic ratio. Phenotypic ratio. Phenotypic ratio means if you look at the plant, you should be able to tell how the plant is from the outside. For example, here we have tall, tall, tall. 3:1. 1 is going to be dwarf. Genotypic ratio. What is going to be the genotypic ratio, students? In the case of genotypic ratio, we have homozygous dominant 1. Then we have heterozygous dominant 2. And we have homozygous recessive 1. So 1:2:1 is the genotypic ratio. Done. Monohybrid cross is done. Monohybrid cross is done now, students. Done. Okay. Now this is your entire monohybrid cross given here in your NCERT. Okay. Now the next concept arises. There is a concept called as back cross. There is a concept called as simple concept, very simple concept called as back cross. Understand with me. Understand. Now there are two types of back cross. There are two types of back cross. Two types of back cross. Okay. Two types of back cross. What are these two types of back cross, students? In the first type of back cross, we take F1 plants and you cross it with dominant parents. Okay, cross it with dominant parents, and such a type of cross is called as outcross. It's called as outcross. In the second type, we take F1 and cross it with recessive parents. Cross it with recessive parents. When we take the F1 plants and we cross it with recessive parents, such type of cross is called as test cross. It is called as test cross. Called as test cross. Clear? Outcross and test cross. Any doubt here, quickly ask me. Any doubt here? Difference between outcross and difference between test cross. Now students, what exactly is a test cross then? What exactly is a test cross? Test cross is a cross. What exactly is a test cross? Test cross, it is a cross between cross between dominant phenotype with dominant phenotype, students, dominant phenotype with recessive parents, with recessive parents, we take the dominant phenotype and we cross it with the recessive parents. But why? The next question is why? Why do we do the back cross, or why do we do the test cross? Why do we do the test cross then? What is the requirement of test cross? It's very simple again, very simple. The main reason we do the test cross, the main reason we are doing test cross is to find the genotype of F1. To find the genotype of the F1. To know the genotype. To know the genotype. To know the genotype of F1 plant. To know the genotype of the F1 plant. Is the F1 plant homozygous dominant? Is the F1 plant heterozygous dominant? If you understood? If you didn't understand, can I quickly teach you? Can I teach you with the help of a diagram now? Can we do the NCERT diagram? Yes. Here we have the NCERT diagram. Students, is this visible or not? Tell me in the chat quickly. Is this diagram visible to all of you? Yes. Tell me. Tell me. Is this visible? Is this visible, students? Is this visible? Visible, yes or no? Quickly. Right? One thing, students, let me drink some water. Let me drink some water, students. Let me drink some water. Once again, all of you drink some water now. All of you quickly drink some water and rehydrate yourself. All of you drink some water and rehydrate yourself. Let me check your attendance. Yes. Chat's a little slow. Yes, chat's a little slow. Okay, one second. F1 plant is there. Then we end up getting okay. Students, let me take some breather. Let me take some breather. Now, students, ready? Ready? Quickly tell me in the chat. Ready? 1 second. Didn't open. You're not marked attendance. My phone is lagging. I'm not able to see attendance also. I'm not able to open this thing also. Students, quickly mark your attendance. I can only see very less attendance. Now, students, what is the test cross? Test cross is basically a cross between your dominant phenotype and recessive parents. Okay, recessive parents. Now listen to me very carefully. Here this dominant phenotype is given. It is violet color flower. Now this violet color flower can be both capital W, or it can be capital W, small w. That is, this violet flower can be homozygous dominant or it can be heterozygous dominant. Right? It can be homozygous dominant or it can be heterozygous dominant. Both chances are there. Now, what do? Obviously, if you take this plant right here, capital W, small w, capital W, this is small w, small w. Small w, small w, this is small w. Right? Small w. Do the simple Punnett square. With the help of Punnett square, if you do, we end up getting all flowers are violet here. All flowers are violet. Why? Because genotype. Because the genotype because the genotype was capital W. If this was capital W, obviously this small w will not be able to be seen anywhere. Now, based on the phenotype which we obtain here, we are able to, based on the phenotype which you observe in F2, based on the phenotype which you observe in F2, we are able to determine the genotype here. For example, in this one, this is heterozygous dominant. So one small w is there. So we end up getting 2 flowers as white, 2 flowers at the violet here. So based on the phenotype which we observe, we are able to get the genotype. Clear? Here, so based on this information, you are able to tell unknown flower is heterozygous here. Why? Because it is producing white flowers. Because genotype itself was a little heterozygous. There one recessive allele was there, and that is why we are getting some white flowers. But in this case here, in the case of your all flowers are violet because the genotype was capital W. Okay, so basically we are looking at the phenotype which we obtain in F2, and we are able to tell the genotype of F1. Is that point clear to all of you? Yes or no in the chat quickly? We are able to determine the genotype of F1 by looking at the phenotype. Okay, understood? Based on what we have the genotype here, we are crossing it and we are looking at the phenotype. We are looking at the phenotype which we obtain in F2, and we are able to determine the genotype. We are able to determine the genotype of F1. Okay, clear? Yes. Now students, now next 3 there are from the monohybrid cross. From the monohybrid cross, there are 2 main laws. From monohybrid cross. The first one is called as law of dominance. The other one is called as, in the chat quickly, the first one is called as law of dominance, and the second one is called as law of purity of gametes. Law of purity of gametes is, what is the other name of it? What is the other name of it? Tell me quickly in the chat. The other name is law of chat is so lagging. I am waiting for the answer. I'm already tired. First one is called as law of dominance. The other one is called as law of purity of gametes. Law of purity of gametes is also called as law of segregation. Thank God, answer came. Also called as law of segregation. Now let's look at law of dominance now. Law of dominance, what does it say? Character are controlled by a discrete unit called as factors. Factors are also called as alleles. Okay, factors occur in pair. We learned alleles are in pair. We learned that. In a dissimilar pair of factors, that is heterozygous condition, one member of a pair dominates. For example, in dissimilar pair of factors, one dominates and the other one is called as recessive. One is dominant, other one is called as recessive here. The law of dominance is used to explain the expression of only one of the parental characters in the monohybrid cross. Where did it go? We'll find here. Of the cross in the F1. See, in the F1, only tall plant was able to express itself. In the heterozygous condition, only tall plant was able to express because capital T is dominant. Capital T is dominant here, right? An expression of both in the F2. It also explains the proportion of 3:1 dominant at the F2. Okay. Now students, That was law of dominance. That was law of dominance. Then what is law of segregation? Law of purity of gametes. It's very simple. Law of segregation basically tells you that the alleles do not blend, they segregate independently. That's all. The alleles, the two different alleles do not blend, and one they will have their individuality. Let me show you the quick example. The quick example is very simple. You have to go to monohybrid cross. Here we have capital T, small t. The two alleles are here. Now this capital T and small t, are they blending? No, they do not show blending. In fact, capital T is individual, small t is individual. Individuality is maintained. Individuality is maintained here. Okay, capital T is separate, small t is separate. Now, why? How can I say they do not show blending? Because why? Because in the F2 generation, the small t is able to express again. Small t is able to express again. So the individuality is maintained. That is called as law of segregation. That is, alleles do not show blending. Alleles do not show blending here. Okay. Alleles do not show blending. Alleles do not show blending. Alleles do not show blending. For example, we have capital T, small t here. They will segregate. And we end up getting gametes here. We end up getting gametes, capital T gamete and small t gamete. Students, it is also called as law of purity of gametes. Why is it called as law of purity gametes? Because this gamete right here, these two gametes right here are pure for a trait. Gametes are pure for a trait. Gametes are pure for that trait. For example, this particular gamete is pure for only tallness. This particular gamete is pure for only dwarfness. So gametes are pure for a trait. Did all of you understand that? That is why it is called as law of purity of gametes. Law of purity of gametes. Each gamete is pure for a trait. And each gamete will have one allele. You can see here, each gamete will have one allele. Each gamete will have one allele. And students, this is universal. There are no exceptions. It is a universal. This particular thing is universally applied. It is going to be universal. But when we come to law of dominance is not universal. There are exceptions in law of dominance. There are exceptions here. Exceptions when we come to the law of dominance. Obviously, the first one is going to be your incomplete dominance and codominance. Incomplete dominance as well as codominance. Incomplete and codominance are the exceptions when you come to law of dominance. But your law of purity of gametes is universal. There are no exceptions at all. Done. Are those points clear to all of you? Quickly tell me in the chat. They are pure for a gamete. They do not show blending. Right? The gametes are pure for a trait. This gamete is pure for a trait. This gamete is pure for a trait here. Okay? They are pure. Now students, the next concept is called as incomplete dominance. Incomplete dominance is a concept in which a particular allele is incompletely dominant over the other allele. So if that is the case, even the recessive allele will be like, even I want to express, even the recessive allele will start to express now. Even the recessive allele will start to express in the case of incomplete dominance. So one allele is incomplete dominant here. One allele is incomplete dominant over the other allele. One allele is incompletely dominant. It is going to be incompletely dominant. Incompletely dominant over the other allele. Over the other allele over the other allele. Now the beauty of this incomplete dominance is, now tell me in the chat, do you think the offspring, will it resemble the parents or will it look different from the parents? Or will it resemble one of its parent or it will resemble both the parents? Tell me. Yes? Tell me quickly in the chat. Quickly in the chat. Students, you need to know the F1. The F1 will not resemble any of the parents. It will not resemble any of the parents. No, I do not want to look like your mother or father. Nothing. It is completely unique. It is going to be completely unique. Examples is dog flower, snapdragon, atrium, starch. You know, the starch synthesis, all the different examples. Dog flower, 4 o'clock plant, mirabilis jellipa. All of these are different examples of incomplete dominance. See, Jai, you need to understand that in examination, if they give you this question, you need to tell universal. Because non-disjunction is a concept which you learn in chromosomal theory of inheritance. Now, obviously, during Mendel time, it was not there. Now we are talking about Mendelian genetics. Under Mendelian genetics, there are no exceptions. Okay? Under Mendelian genetics, there are no exceptions to law of purity of gametes. Okay? Understand that. So in examination, if they give you, you should able to tell that yes, it is universal. You cannot tell non-disjunction and all. Okay? Mirabilis jalapa, dog flower, snapdragon, starch synthesis, all of these are different examples of incomplete dominance. So what exactly happens in incomplete dominance, students? What exactly happens in incomplete dominance? Very simple. First of all, let's write the parents here. Let's write the parents. We have the parents here. One is red flower, one is going to be your white flower. Red flower and white flower. What are the gametes here? Gamete is going to be your, this is capital R. This is going to be capital R, small r. Or no, capital R, small r only, right? Small r only. Yes, capital R, small r. Now here we get single gamete. If we get single gamete, this is your gametes. Now we do crossing over, we end up getting the F1. F1 is what? Capital R, small r. Now technically, students, if this was normal, a normal cross, this would have been red. Normal cross red. Since it is incomplete dominance, this particular flower will be pink in color. It's going to be pink in color. Now let's do selfing of F1. Selfing of F1. That is capital R small r into capital R small r. Okay, selfing of F1. Okay, let's do it here. Down, Punnett square. Capital R small r, capital R small r. What do we get? Now, tell me the result now. Tell me the phenotypic ratio and genotypic ratio now. What is going to be the phenotypic ratio and genotypic ratio? First we'll do phenotypic ratio. Go. This one will be completely red. These two will be pink and this will be white. So, one red, two pink and one white. That is phenotypic ratio. What is genotypic ratio then? Genotypic ratio, homozygous dominant 1, heterozygous dominant 2, homozygous recessive 1. So 1:2:1, and this is the case in which your phenotypic ratio is equal to genotypic ratio right here. Done. Phenotypic ratio is equal to genotypic ratio here, students. Done. That is going to be your monohabit cause. That is going to be your incomplete dominance. Now there is something called as concept of dominance. Now tell me honestly, how many of you have read this part and understood this part very well? What is the meaning of concept of dominance? Just be honest with me. Just be honest. What is the concept of dominance? Concept of dominance basically tells you. Concept of dominance basically tell you what is this dominant, what is the meaning of dominant, and what is the meaning of recessive. What is the meaning of recessive? Okay, that is what it tells you. That is what it tells you. Very simple. It's very simple. It's very, very simple. Okay. Yes. Which one you trying? Sony, what is the doubt? I think I missed the doubt. Yes. Red is dominant. Why white? NCERT says white. Red is always dominant. More attracting the flowers. Attraction. With the help of evolution also you can say red attracts more flowers. White is always recessive. New NCERT have changed, no? I have to check the new NCERT then. In NCERT, R smaller is given as dominance. What are you saying? Scamming me, huh? Trying to scam you something. What are you saying? Yeah. Oh, here I've written small. 1 second. Okay, this one I think I wrote a mistake here. Yeah. Then you only get a single gamete. You get a single gamete. Okay. Is this clear now? Okay. I think I was just writing it and I just went forward. It's recessive. Obviously, it's homozygous recessive. It is supposed to be homozygous recessive. Homozygous recessive. Homozygous recessive. Understood? Clear? Yes. Understood. Yes, understood. It is homozygous. See, here when I was writing it, I just wrote it like by mistake, but yeah. Smaller, we get pink color flower. This is red and this is white. Smaller. Happens. Okay. Now, what is the concept of dominance? What is this concept of dominance, students? What is this concept of dominance? Now, here we have a gene, students. We have a gene here. Okay. We have a gene here. Now, this gene will end up giving us some protein, and this protein will express. This protein will express eventually. So all of you know a gene has 2 alleles. Yes, it has 2 different alleles. Okay, 2 different alleles. Now students, if you look at it only, you'll be able to tell there is a clear difference between dominant allele and recessive allele. Yes, there's a difference. There is a difference here. That is, can I tell small t is gonna be a little different from capital T? Yes, small t is going to be different than capital T. So can I tell small t is a different? The main reason for difference could be some form of mutation. Yes, its difference could be because of some form of mutation. Yes, it could be some form of mutation, and it can be different. Yes, Sony is the topper. Yes, it could be because of some form of mutation, right? It could be some form of mutation. Yes. Now, here we have a scenario. Students. Here we have a scenario. Imagine a gene is producing an enzyme. Enzyme. Now, this enzyme can be of 3 types. The first type of enzyme, if this gene is producing an enzyme, it could be normal enzyme, or sometimes normal, but it is less efficient. It could be less efficient. Yes, normal enzyme. Other enzyme can be non-functional. Non-functional. Yes, other enzyme can be non-functional, or sometimes there can be no enzyme at all, like enzyme is completely absent. Enzyme is absent. Now students, this concept of dominance and recessive, this concept of dominance and recessive, it originated from something called as mutation. Now here, when the enzyme is normal or less efficient, it is dominant, and these two cases it is recessive. It is recessive in nature. So students, if you want to understand what is, that's why I told you, right? That's why I told you before. The dominant is also called as wild type, and recessive is also called as what? Modified type. It is also called as what? Modified type. That's all is the concept. That's all is the concept here. Okay, so no enzyme is absent or no enzyme at all, non-functional, recessive enzyme, or modified. That is, the small t gene has been modified or mutated. It has been mutated, right? Then we have normal or less efficient, that is going to be your wild type. Understood? That is the concept of dominance. The concept of dominance originated because of some form of changes. Yes, sweetie. Right. So that is the concept of dominance. That's all. Now next concept is codominance. Students, this chapter is very big. Questions can come from anywhere, questions can come pop up. Okay. Now this is the first part where we'll be talking till chromosomal theory here of inheritance. Till there I'll talk. Okay. Unmodified means normal. Simple, opposite the following it is, right? We'll be doing today with chromosomal inheritance. After chromosomal theory of inheritance, I'll be teaching you linkage in the next class. Okay, so we'll go step by step. If I teach you for 4-hour class today only, won't you feel little fed up? Tell me honestly. You'll feel little, cooked up. So I want you to, whatever I'm teaching right now, learn what I'm teaching, and after the class, I want you to read the NCERT once and try to solve some questions. Okay, that is going to be the entire flow. Now, codominance. In codominance, what exactly happens? In the case of codominance, both the alleles are dominant. Here. Both the alleles are dominant here. Okay. Now, what about the offspring? Will the offspring look like a dominant allele or the recessive allele, or father or mother, basically? How will it look like? Tell me. The offspring will not resemble either it look like both the parents. The offspring will resemble both the parents. It will resemble both the parents. Okay, both the parents. That is going to be your codominance. To understand codominance, there is a concept called as multiple allelism. Multiple allelism. Now, what is this multiple allelism, students? Multiple allelism is a simple concept which says that one gene can have multiple alleles. One gene can have multiple alleles. And one such example is going to be your ABO blood grouping. Why? Because in the case of ABO blood grouping, we have 3 different alleles. We have 3 different alleles in the case of ABO blood grouping. In the case of humans, we have 3 different alleles. That is IA, we have IA, IB, and I0 or I. Now these two IA and IB are dominant alleles. They are the dominant alleles, and I0 or I is called as the recessive allele. It is called as the recessive allele. Now students, in the case of humans, you cannot have all 3 alleles. Out of the 3 alleles, only 2 will be present. only 2 will be present. Only 2 will be present here. Okay, out of the 3 alleles, only 2 will be present in the case of your humans. In the case of humans. Okay, now let's take one quick example. Tell me your blood group in the chat quickly. Tell me your blood group in the chat. What is your blood group? What is going to be your blood group? Quickly tell me in the chat. What is your blood group? What is your blood group? Quickly in the chat. What is going to be your blood group? A, B, O, O positive, A positive, B positive. Positive, negative, that is RH factor, which we will discuss later on, but that is not in the syllabus right now. Just tell me O, A, B. Okay, let's take Ravindrani. She's become very active today. Okay, for example, Ravindrani blood group is B, B positive. Forget the positive right now, just learn the B. Just learn the B, okay. Now, what could be the possibilities of the parents? What could be the possibility of parents? Okay, parents possibility. Parents can be IO and IB. Father can, this is father, this is father, this is mother. Okay, other thing can be, both can be IB, IB. Yes, both of them can be IB, IB. The other thing is, father is IB and mother is I naught. Father is I, mother is I naught, right? So in this case here, this is going to be dominant, so A blood grouping. Here both are dominant, so B blood group. Here B is dominant, B blood group. Did you realize that is called as blood grouping? That is called as blood grouping, and you will get a lot of questions. You will get a lot and lots of questions from the blood grouping. That is very sure. That is very, very sure. Okay. Now here you need to understand, how does the blood grouping actually work? How does the blood grouping actually work? The blood grouping, the main reason for the different types of blood group is because of the sugar present here. Because of the sugar present here. For example, students, let's take two different cells here. These are red blood cells. On these red blood cells, there is something called as H antigen, H plus antigen or H antigen, H antigen. Here also we have H antigen, H antigen. Now along with H antigen, along with this H antigen, there is a sugar molecule. There is a sugar. There is a sugar. Okay. Along with H antigen, there is a sugar. The first type of sugar is galactose in the case of your B blood group. There is a sugar called as galactose. And when the galactose is present along with H antigen, it has become B blood group. Now what about in the case of A? What about in the case of A, students? In the case of A, the sugar is called as N-acetyl. N-acetyl galactosamine. N-acetyl galactosamine, and this becomes A blood grouping. It becomes A blood grouping. Now what about in the case of O? What about in the case of O? In the case of O, there is no sugar. There is no sugar. That is why it's called as I naught. That is why it is called as I naught. So the main difference between A and B is the slight change in the sugar. Slight change in the sugar here. That's all. Okay, slight change in the sugar. Now here, if you look at this paragraph, this beautiful graph here or table here, if parent has IA, so blood grouping is going to be IAA, so A. IA and IB both are dominant, so IA and IB blood group is going to be AB. One parent is IA, other parent is small I, so IA is dominant, so we end up getting blood grouping A. So based on that, you can tell the different types of blood group. Different types of blood group. Okay, this is very easy to understand now. Now students, now we will do dihybrid cross. Now we will do the simple dihybrid cross. Can we start now, students? Tell me in the chat. Students, the class is going very smooth. Do you realize 1.5 hour is over? 1.5 hour is over, and we are going in a very good pace, and in the next 20 minutes the class will get over now. Next 20 minutes the class is going to get over, and you can go free and run around. Okay. Go free and run around after that. And next class is going to be in the next coming week, where we'll be doing the cell part 2 and this chapter part 2. This chapter will have also a part 3, remember that. This chapter will also have a part 3. In part 3, I will only be doing numericals. as requested. Okay, we'll be doing only numericals as requested because I know numericals can become a little difficult because all of us love biology and hate maths, right? All of us hate maths. So numericals, separate session. What's that? Okay, separation for numericals. Now, inheritance of two genes, inheritance of two different genes. Okay, what exactly happens now? What are the parents now? First parents, selection of parents. Selection of parents. Parents is very simple. Capital R, capital R, capital Y, capital Y. That is going to be round and yellow. This is right here is round and yellow. It's going to be round and yellow. The other parent is going to be small r, small r, small y, small y. What is this going to be? This is going to be wrinkled and green. It's going to be wrinkled and green. Wrinkled and green. Okay, wrinkled and green. Now students, right, almost 1.5 hours is completed, right? Now we do crossing. Okay, what do we get in the cross, students? We do crossing and we get capital R, small r, capital Y, small y. Now what is the phenotype here? The phenotype is very simple. Capital R is dominant, so round. Small y, capital Y is dominant, so yellow. It's going to be round and yellow in the case of F1 progeny. In the case of F1. Okay. Now what is the next step? Selfing of F1. Next thing is going to be selfing of F1. Now what do we do in selfing of F1? First of all, we need to know the art of making gametes here. For example, here we have capital R and capital Y, small y. How do you make the gametes here? How do you make the gametes here? Very simple. Cross with this and this with this. This with this and this with this. So we end up getting capital R, capital Y, capital R, small y, small r, capital Y, small r and small y. These are the 4 different gametes which you get. Sir, yes? I know. You understand? This is how you end up getting the gametes. Now all you need to do is again the Punnett square. Again do the Punnett square. Here also you write capital R, capital Y, capital R, small y, small r, capital Y, and small r and small y. Then you do the simple cross, students. I will not do the entire cross. I'll just show you one. How do you do this? Capital R, capital Y, small y. This one, capital R, capital Y, small y. What about this one? Capital R, small r, capital Y. Last one, capital R, small r, and capital Y and small y. Similarly, students, you need to do the entire Punnett square. Once you do and you find the phenotypic ratio in the chat, go. What is going to be the phenotypic ratio now? What is going to be the phenotypic ratio? The total progeny, first of all. Total progenies is going to be 16 here. 16. 1, 2, 3, 4, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. 16 progenies, right? This is going to be 16 progenies. How will be the distribution? Distribution is very simple. Distribution is going to be, the ratio is 9:3:3:1. 9:3:3:1 is the ratio. Okay. 9:3:1 is the ratio. Now here, round in yellow. Round in yellow is going to be 9. Round in yellow is going to be 9. Wrinkled in yellow is going to be 3. Wrinkled and yellow is going to be 3. Round yellow, wrinkled yellow. Wrinkled yellow. Now round in green. Round and green. And last one is going to be wrinkled and green. Wrinkled and green. Out students, one more thing which is very important, all the questions you can solve here, all the questions you'll be able to solve here is with the help of, you need to understand these two are parental. Parent was round in yellow, dominant. Wrinkled in green, parent. These two, can you see these two are recombinant. These two are recombinant type. They are the recombinant type. Wrinkled in yellow and round in green are going to be your recombinant type. This is phenotypic ratio. But how do you make the genotypic ratio, students? How do you find the genotypic ratio? Very simple trick. I will teach you the trick here. Listen to me very carefully. Even if you do not know monohybrid, even if you do not know the phenotypic ratio of dihybrid, for that also have a trick. You need to understand your dihybrid cross. Your dihybrid cross is basically two monohybrid crosses. Two monohybrid crosses. Example, listen to me. Listen. For example, the phenotypic ratio. Tell me the phenotypic ratio in the case of monohybrid. 3:1 and 3:1. In the case of monohybrid phenotypic ratio, do the multiplication. 3 into 3 is 9. 3 1. This into this, this into this, this into this, this into this. Okay. 3 into 3 is 9. 3 1s are 3. 3 into 3, 3. 3 into 1, 1. 9 is to 3 is to 3 is to 1. Done. 9 is to 3 is to 3 is to 1. Now, how do you find for the dihybrid? Just tell me the genotypic ratio in the case of monohybrid. 1 is to 2 is to 1. 1 is to 2 is to 1 was the genotypic ratio in the case of monohybrid. Now, cross it. 1 into 1 is 1. 1 into 2 is 2. 1 into 1 is 1. 2 into 1 is 2. 2 2 is 4. 2 into 1 is 2. 1 into 1 is 1. 1 into 2 is 2. 1 into 1 is 1. This right here, students. 1 is to 2 is to 1 is 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. That right here is the genotypic ratio. Is going to be your genotypic ratio of dihybrid. Understood? Understood, students? Understood? Nice. That is going to be your dihybrid. Okay. Inheritance of two genes. Now, with that, you end up getting with the help of monohybrid cross, we got law of dominance as well as law of segregation. Now, with the help of, right? Now, for the law of, with the help of dihybrid cross, what do we get? With the help of dihybrid cross, there is a simple law. The simple law says that is called a law of independent assortment. Law of independent assortment says that when two pairs of contrasting traits, two pairs of traits, here we had seed shape and seed color. Here we had seed shape, Can you see? Round and yellow is the seed shape and seed color. Seed shape and seed color. When a pair of contrasting traits, students, when a pair of contrasting traits are combined in a hybrid, segregation of one pair of character, segregation of shape of the seed, segregation of color of the seed is independent of other pair characters. That is, color of the seed, students, color of the seed and shape of the seed. Color of the seed and shape of the seed, they do not mix with each other. Each character is always separating individually. They are separating individually. Is that point clear to all of you? Yes or no in the chat quickly? Is that point clear to all of you? Tell me in the chat. Is that point clear to all of you? Clear? Clear? Easy. Just checking the chat. Here the chat is lagging a lot today. Sony Mohan, pleiotropy, I'll tell you, it'll come. It's called as mummy genes. I call pleiotropy as mummy genes. And pleiotropy is basically happens where one gene has multiple phenotypic effects. One gene has multiple phenotypic effects. Right? We'll get to that. We'll get to that. We'll one by one will come. Okay, don't worry. Why is the chat so slow today also? Here also chat is lagging. The phone also chat is lagging big time. Okay, don't worry. Okay, now we'll go to the next concept now. That is going to be rediscovery of Mendel's work. Rediscovery of Mendel's work. Now I told you Mendel's work was not very recognized when he was alive. Only when he passed away all his work was recognized. That is, Mendel published his work in 1865 but remained unrecognized till 1900s. See, even after his death, his work was not recognized at all. Not recognized at all. That is, probable causes, work could not be widely published. Factors were stated as stable and discrete units by Mendel, and Mendel's concept of alleles never blend was not accepted by his contemporaries. They did not accept his theories at all. Right? Use of mathematics to explain biological phenomena was not unacceptable. They're like, no, maths not allowed here. Maths not allowed here. No physical proof of existence of factors because we were not able to look at the factors. Obviously, alleles but technology was very behind. Right? There was no physical proof. So his work was not recognized. Only when there were three different scientists who recognized his work were Hugo de Veres, Karl Konz and Heinrich von Schirmack. These three different scientists recognized his work later in his life. Once Mendel was gone, his work was recognized again. Three different scientists, students. And with that, there are two more scientists. Now, what are these two more scientists? The two more scientists is called as your Sutton and Boveri. There are two more scientists called as Sutton and Theodore Boveri. The two different scientists in the year 1902. In the year 1902, they gave the chromosomal theory of inheritance. Now, this chromosomal theory of inheritance is very simple and very easy. Because chromosomal theory of inheritance tells you what? Chromosomal theory of inheritance is telling you only one thing. Chromosomal theory of inheritance is telling you that there is a parallel relationship between, that is, behavior of a chromosome and behavior of a gene is same to same. That's all. Behavior of a chromosome and behavior of a gene is same to same. If chromosome is splitting, even gene is splitting. Why? Because technically gene is present on the chromosome. Right? So if the chromosomes are splitting, even the genes are splitting. That is chromosomal theory of inheritance. And they were able to explain chromosomal theory of inheritance with the help of Mendel's work. Can you see here? They observed that behavior of a chromosome is parallel to the behavior of a gene. Same function is there, right? Use chromosome movement. They use chromosome movement. How chromosomes move during cell division to explain Mendel's law of inheritance. So they were able to explain Mendel's law of inheritance based on the chromosome movement during cell division. Understood? Same to same. Chromosome movement is same and gene behavior is same. Both of them are same to same. Technically they are same only because gene is present on the chromosome. Right? Is present on the chromosome. Yes, ma'am. Come. I was teaching them chromosomal theory of inheritance. We have class now, ma'am.
I have DNA.
Now?
No, in some time.
2 o'clock, right? 2 o'clock.
Hello.
Tell them hi, ma'am.
Hi.
Yeah. No, they're telling early morning. Some of the students are like that we were not available here to take the classes.
Who is not available?
Students, because they have to go to school.
Oh, appadi. You don't have.
Okay, never mind. Yeah.
Students will understood. Appadi. Where's the Josh? Yeah, tell hi. No.
Yeah.
What happened? Sleeping?
No, there is a major lag now. There is a major lag.
Okay.
No, usually the lag is like 5 seconds. Today is 10 seconds. I don't know why. Latency would have kept low.
Now.
Now, after 10 to 15 seconds, the
It'll be so funny that we are staring and they'll be like, why are they staring and not telling us hi?
Exactly.
So students, now the messages are coming, now I'm able to
The
see.
funny part is I asked them a question and I'm waiting for 10-15 seconds. I'm like, answer is not coming. I'm like, do they not know the answer or am I not audible to them? It is such a,
I know. Awesome. Very nice. Students, tomorrow my NCERT line by line is there, not today. Okay?
Tell them the time also.
Yeah, time is 6 p.m. Okay. NCERT line by
Now
line.
you don't have excuse that time. You don't have an excuse to tell that no, I will
was
be
in school.
Some tuition or something. You have to
Yeah.
attend.
Exactly. So
Yeah.
tomorrow at 6 p.m. you will have NCERT line by line by your own Gopika ma'am. Biological classification we will be doing. Okay. So yes, go and also mark the attendance there. Do not forget and mark the attendance in this video also. What are you people waiting for?
Yeah.
Next blue moon they will.
Then they'll come again. They'll see the video. Oh, I forgot to mark attendance. Then they'll attendance.
Yes. Okay.
Yeah.
See you tomorrow. I want to see all these names tomorrow.
Yeah.
Okay. Hi Rishit. Okay. Hi. Okay, sir. Continue. Bye bye.
Bye, ma'am.
I'll meet my DNA students.
Bye, ma'am. So ma'am is gone and ma'am is, see students, do you realize that we come to office, we take multiple paid batches, we take on YouTube and we always make sure that YouTube is not compromised because we are teaching somewhere else. So we always make sure the entire content is produced on YouTube also as well as on the paid batches. We don't show compromise anywhere. Just because we are teaching there, we don't stop here. You should know that, right? Now students, what are the differences? See, similarities between your genes as well as your chromosomes. What are the dissimilarities? Chromosomes occur in pair. All of you know homologous chromosomes, and your genes also occur in pair, like factors, alleles are in pair. Yes, factors in pairs. Chromosomes segregate during gamete formation, and even genes segregate during the gamete formation. Yes, alleles separate during gamete formation. Factors segregate during gamete formation. Yes, chromosomes segregate independent of each other. Factors segregate independent of each other. We learned independent assortment. So there is a similarity between behavior of chromosome as well as behavior of the genes. You should know that, right? You should know that. Now students, this is a diagram just given you NCERT. That it basically is showing you the separation of chromosomes. See, here we have chromosome at G1. We have chromosome at G2. Yes, at G2. Why? Because the S phase will come in the middle. Then we have meiosis 1 anaphase. Now tell me what happens in anaphase? Separation of chromosomes or separation of sister chromatids? Tell me. In the case of anaphase 1, there is separation of homologous chromosomes. Yes, separation of homologous chromosomes. Yes. Then we have anaphase 2. In anaphase, there is separation of sister chromatids. Then we have the germ cell. So students, you need to understand the separation of chromosomes is similar to the separation of genes. That's all. Separation of genes. Now here also, see here, the only purpose of this entire figure is to show you that yes, in the case of chromosomes, there is crossing over. Yes, in the case of chromosomes, there is a crossing over, and also in the case of your, Mendelian crosses, we have crossing over. For example, we have capital R, capital Y, small y. We have shape of the seed and color of the seed here, crossing over. Here also we can see crossing over, same to same thing. Okay, same to same thing. Now students, this is the last concept for today. And who gave the experimental proof? Experimental proof for the chromosomal theory of inheritance was given by none other than T.H. Morgan. It is given by T.H. Morgan, and his main work was on Drosophila. That is a fruit fly. Now after this concept, there is something called as linkage and recombination. In Hindi, it's very difficult, Ayush. I can speak in, I can speak in Hindi. हिंदी में बात कर सकता हूँ, पर हिंदी में पढ़ा नहीं सकता. बहुत दिक्कत आती है. See, that is a difficulty. I can speak in Hindi, but after some time it becomes very difficult to talk. The tongue is not adjusted to it. No, the tongue is not adjusted to talking in Hindi. That's a difference, right? So T.H. Morgan. Now there is a student of T.H. Morgan. What is linkage, recombination? All that can we do in the next class. all that can be doing in the next class now. Now that's all is the today's class is going to be. And last concept for today, before we go, the last concept for today is going to be why fruit flies. The concept is going to be why fruit flies. Why fruit flies? Like there should be some reason. No, he has taken fruit fly. I told you like Mendel has taken pea plant. Mendel has taken a pea plant. So your T.H. Morgan has taken fruit flies. Why? Again, right? Why? Why fruit flies? Right? Very simple. Drosophila melanogaster, which we found on the suitable for the studies, they could be grown. First point here, they could be grown on the simple synthetic medium in the laboratory. They can be grown very easily. Yes, very easily they can be grown. Yes, they complete their life cycle. Point number 2. They can complete their life cycle in about 2 weeks. Point number 2. A single mating could produce a large number of progenies. Point number 3. There was a clear differentiation of the sexes. Point number 4. That is, male and female can be distinguished very easily. Yes, distinguished very easily. Yes, that is the 4th point. Now, then 5th point is has many types of heredity variations that can be seen with the low powered microscope. That is eye color, body color, wing color, all that can be seen with the help of simple microscope. And students, that is the reason, that is the main reason why T.H. Morgan took the fruit flies. Done? Done, students? And that is for today's class. Now you might be wondering, students, do you need to solve questions? Yes, you need to solve questions. Now students, if you are looking for a good test series, right, if you are looking for a good test series and you need to find a good test series, if you go to the link in the description right now, you will find the best test series in the India. And this test series, students, you will have long test, you will have part test, you will also have subject wise test, you know. So if you're looking for a good test series, if you want to join year long, not just one, year long test series, description it is there, go check it out once. Okay, so students, that's it for today's class. Thank you so much, and I will see you in the next class. And the next class is going to be line by line. Next class is going to be line by line. After the line by line class, we will do the inheritance. Okay, questions not now, questions not now. Questions I want to keep separately. Ravindra, you should understand, numericals will be so many numericals, right? So many numericals is going to be there. So questions, a separate class for a session. We will keep a separate 1.5 hour or 2 hour class only for questions. I will bring from entire internet everywhere. I will find questions and I will bring for all of you. Okay, I will bring all types of numericals for you. We will be doing every single PYQ for you in a separate numerical class. Okay, so we'll do that. Take care, students, and do not forget to let me know in the comment section how was today's class. That is very important. Okay, let me know in the comments how was today's beautiful class. It was a very chill class, and I hopefully all of you understood what I thought, right? And if you have any doubt, let me know in the comment section and I will be glad to clear all the confusions. Okay, so thank you so much, students, for attending today's class, and I will see you in my next class now. Take care, all of you. Bye bye. Bye bye. Take care, all of you. Take care.